we have discussed with each other on the eukaryotic genomes. And we know that in the eukaryotic genomes, we side of the nucleus genome, which is a liner DNA, which is the DNA associated with the histone protein to form chromosome. We have another genomes located in the mitochondria, and in case plant cell, also the chloroplast. And the scientists have provided a theory called the endosymbiotic theory, in which million years ago, prokaryotic cell had been symbiotic with the eukaryotic cell, get inside and participate in the local glycolysis to synthesis the energy for the host cell. And the evidence they provide for this theory is the genomes of the mitochondria is similar to the Asian prokaryotic cells. So today we will explore one example of the mitochondrial genomes is the human mitochondrial genomes. So the first human mitochondrial genome was sequenced on 1999. So until now, there's many different other versions of the human mitochondrial genomes have been published. So to explore the human mitochondrial news, human mitochondrial genomes, you can go on Google. Wait, I can share my screen. Okay. So you can go on Google and type human mitochondrial genomes. And you click on the link mitomap.org. Okay. So in here, you can click on the annotated human mitochondrial DNA sequence. So this is the newest human mitochondrial genome called RCRS, or Revised Cambridge Reference Sequences. So in here, they have provided all the sequence of the genomes. Here, 16, more than 16 kilobase of the human mitochondrial genomes. And also annotated of different regions. So the annotated, that means the explanation which is the meaning of each region of the genome is in here. So for example, we have the D loop here is we have two regions of the D loop. First is from 16,000 to 16,569, and the other is from the beginning to the position of 576. So we have different genes here. So the next one from 577 to 647, which is relatively small, just less than 100 base pair, is the gene TRNF, which is encoding for the tRNA phenylalanine. Okay, so based on the review here, we can see that the human mitochondrial genomes was contain 37 genes, in which they coding for 2 RNA, 2 ribosome subunit RNA, 22 tRNA, and 13 polypeptide. So this is the region of the different the map of the human mitochondrial genomes. So the first, yes. Okay. So if anyone want to send me a message, please send it us to everyone, because maybe the problem is everyone concerning. So the first region is the D-loop region. We also call the hypervariant. Okay. So in here, this is the region not coding for any protein. It just regions coding for nothing, but is varying between individuals. So they use them to categorize which individuals, the identity of each individual. So the human mitochondrial genomes have several advantages as a molecular marker compared to the nucleus genomes. First, it is mostly maternal inherited. That means that you will inherit most of the mtDNA from your mother, and your mother will inherit most of the mtDNA from her mother, and so on. Her mother is inherited the mtDNA from her mothers. So it's easier to track the source of the mtDNA compared to nucleus genome when it's a combination between a half is from paternal and a half is maternal. Secondly, the human mitochondrial genomes, as you can see, is a circular DNA, so it's quite stable, more stable than the linear DNA, yes. So this technique has been used to identify the identity of our soldier who sacrificed during the war in the war against the American. So during the war, many of our soldiers were sacrificed and they was buried in hurry without any coffin. So under the effect of the weather, of the UV light, of the rains, so most of the nucleus DNA have been damaged, but the mitochondrial DNA is quite stable, so it still remains in the bone, in the teeth. So based on this DNA fragment, scientists can, based on this, to identify, to compare with the genetic background from the mother side of the soldier to make sure that he belongs to the family. So first, let's watch a small animation to have an overview about the structure of the human mitochondrial DNA.
Mitochondria, found in all cells with nuclei, release energy from food, and store it in ATP molecules. Mitochondria carry its own DNA. Mitochondria were once free-living bacteria that were engulfed by ancestor of eukaryotes, but not digested. Bacteria and larger cell established a symbiotic relationship, in which the larger cell provided protection, while bacteria produced food. The mitochondrial chromosome preserves many features like bacteria. First, they are circular molecule. Human mitochondrial chromosome is only 16,549 base pairs long, which is not much larger than any bacterial plasmid. Second, the mitochondrial chromosome is tightly packed with genes. Unlike nuclear chromosomes, which have large intergenic regions of non-coding DNA between genes. Third, most mitochondrial genes lack introns, which are non-coding information within nuclear genes. Human mitochondrial DNA contains 37 genes. 13 genes are involved in process of oxidative phosphorylation. 22 genes encodes transfer RNAs for specific amino acids, whereas 2 genes encodes 2 subunit rRNAs of ribosomes. Control region. The control region contains the signals to control RNA and DNA synthesis in mitochondria. It is also our fastest evolving DNA sequence. This hypervariable region accumulates mutations at approximately 10 times the rate of nuclear DNA. This region is also called D-loop, relating to structure formed when mitochondrial DNA replicates. Complex 1 genes. These 7 genes encode subunits of NADH dehydrogenase, which is an electron transporter, involved in production and storage of energy. Though the genes lie in mitochondrial DNA, the process of producing energy requires molecules that are produced outside of mitochondria. Complex 3 genes. This mitochondrial gene encodes cytochrome B protein, which is part of inner membrane of mitochondria, known as ubiquinol, or cytochrome C oxidoreductase. Complex 4 genes. These 3 genes encode subunits, which are part of cytochrome C oxidase complex. This complex acts as electron transporter during process of producing and processing energy in mitochondria. Complex 5 genes. These 2 genes encodes proteins of ATP synthase complex. This complex is located in inner membrane of mitochondria. Ribosomal RNA genes. These 2 genes known as 16S and 12S are RNA, which are used to build ribosomes. During protein synthesis, ribosomes assemble to translate messenger RNA. Transfer RNA genes. These 22 genes encodes transfer RNA molecules. Each transfer RNA is associated with a specific amino acid. Transfer RNA plays an important role in protein synthesis, delivering their associated amino acid to ribosome for incorporation into new protein. The human mitochondrial chromosome has been vastly reduced. Over time, genes for functions that could be provided by the host were lost. Also, some genes needed for respiration were transferred to nucleus. Millions of years of evolution have resulted in small mitochondrial chromosomes. When egg and sperm unite, the sperm's mitochondria are discarded. Mitochondrial DNA is therefore inherited only from the mother. Within a family, females from grandmother to daughter all share identical mitochondrial DNA. Unlike DNA in nucleus, mitochondrial DNA does not recombine with other chromosomes.
Ok, mình sẽ giải thích lại một chút bằng tiếng Việt cho bạn nào vẫn còn đang là băn khoăn về cái phần này. Thì có thể là cái phần này đang hơi mới với các bạn. Như buổi trước mình đã trình bày rồi là ở các tế bào eukaryotic thì bên cạnh cái hệ nhân typically nhân ở hệ genome ở trong nhân thì nó được xem như là có một cái hệ genome nữa ở trong ti thể và ở các tế bào thực vật là lục lạp. Bởi vì là ở các bào quan ti thể và lục lạp, các nhà khoa học phát hiện ra là các bào quan này cũng có hệ DNA riêng của nó. Thế thì người ta đã đưa ra một cái giả thuyết là cách đây hàng tỷ năm thì có một cái sự cộng sinh đã diễn ra và các vi sinh vật nhân sơ có prokaryotic cell bằng cách nào đó đã chui vào bên trong cái sinh vật nhân thực eukaryotic cell để làm nhiệm vụ là tham gia vào quá trình glycolysis và tổng hợp nên ATP, có nghĩa là năng lượng cho các cái tế bào này. Bởi vì là người ta đưa ra cái giả thuyết này bởi vì dựa trên cái bằng chứng là khi người ta giải trình tự cái hệ genome của cái cơ quan ti thể, người ta thấy là cái hệ genome này có rất nhiều điểm tương đồng so với các loài vi khuẩn cổ. Dựa trên đấy người ta đưa ra một cái giả thuyết như vậy là các cái bào quan này xuất phát là những cái loài vi khuẩn sống độc lập, và sau đó thì do điều kiện tác động của môi trường như thế nào đó thì nó đã chui vào bên trong tế bào eukaryotic cell để làm nhiệm vụ cộng sinh. Thì các eukaryotic cell sẽ cung cấp nguyên vật liệu cho các cái mitochondria để làm nhiệm vụ tổng hợp năng lượng. Thì ở đây mình đã đi sâu vào một cái ví dụ là cụ thể là cái hệ DNA ti thể ở người. Thì hệ DNA ti thể ở người thì đầu tiên được công bố giải trình tự vào năm 1999 với kích thước là khoảng độ hơn 16 KB, cụ thể là 16.569 base pair, và mã hóa cho 37 gen, trong đó thì 2 gen là mã hóa cho các tiểu phần ribosome RNA, 22 gen là mã hóa cho các tRNA là RNA vận chuyển, và 13 gen là mã hóa cho các polypeptide. Các polypeptide này thì chủ yếu là tham gia vào quá trình glycolysis. Thế thì vùng duy nhất mà không mang mã, bởi vì là DNA từ vi khuẩn cho nên hầu hết là các đoạn DNA là mang mã, nhưng mà vùng duy nhất mà không mã hóa cho cái gì cả là cái vùng đi loop này. Và cái vùng đi loop này thì còn được có tên gọi là hypervariant, dịch ra tiếng Việt gọi là vùng siêu biến. Tại sao gọi là vùng siêu biến? Bởi vì là cái vùng này nó rất là khác nhau giữa người này với người kia. Thế thì người ta dựa trên cái sự khác nhau đấy để người ta làm cái gọi là chẩn đoán, gọi là định danh ADN. Ví dụ như là một cái công nghệ định danh DNA dựa trên cái đoạn đi loop này là Viện Công nghệ Sinh học đã mở ra một cái trung tâm gọi là Trung tâm giám định hài cốt liệt sĩ. Thì các bạn biết là ngày xưa kháng chiến chống Mỹ thì các liệt sĩ chúng ta hy sinh là hầu hết là chôn cất rất là vội vã và không có áo quan, cho nên là dưới cái tác động của nắng mưa gió, các điều kiện khí hậu môi trường thì hầu như cái DNA ở trong nhân là nó bị phân rã hết rồi. Nhưng mà DNA ở trong thi thể thì nó có một số lợi điểm để mà có thể sử dụng như là một cái công cụ định danh rất là hữu ích. Thứ nhất là nó rất là bền, bởi vì DNA là DNA mạch vòng, không phải là DNA mạch thẳng, cho nên nó bền hơn so với DNA mạch thẳng. Thứ hai là hầu hết DNA ti thể thì nó lại di truyền theo dòng mẹ, có nghĩa là mặc dù một số công trình nghiên cứu gần đây chỉ ra rằng là chúng ta có thừa hưởng một ít ti thể từ bố nữa, nhưng mà hầu hết đến hơn 95% ti thể của chúng ta là hầu hết là di truyền theo dòng mẹ. Mẹ thì lại thừa hưởng từ bà ngoại, đúng không ạ? Bà ngoại thì lại thừa hưởng của mẹ của bà ngoại, cho nên là cái việc mà lần giấu theo cái DNA hệ ti thể này thì nó rất là tiện lợi, vì là DNA ti thể thì nó không có trao đổi chéo mà hầu hết là chỉ là theo một dòng thôi, chứ còn DNA trong nhân là một nửa từ bố một nửa từ mẹ, cho nhiều khi là truy xuất nguồn gốc là hơi khó. Cho nên là cái DNA ti thể này đã được sử dụng rất là hữu ích trong cái công việc là định danh các thầy cốt liệt sĩ, vì là chỉ dựa trên một mẩu răng hoặc một mảnh xương của các thời kỹ liệt sĩ thì hiện giờ Bộ Lao động và Thương binh Xã hội đã có một cái chuyên đề riêng dành cho Viện Công nghệ Sinh học, thành lập một phòng riêng, phòng DNA ứng dụng để ứng dụng cái công nghệ này. Cái công nghệ này thì đầu tiên là được các nhà khoa học nước ngoài sử dụng để định danh những cái xác ướp mà được phát hiện đã chết cách đây hàng ngàn năm rồi, vẫn còn có thể sử dụng được để có thể định danh. Okay. So here I have made a small comparison on the differences in the genome, human nuclear genomes and the mitochondrial genomes for you. And this is one important part that you have to remember if you remember in the survival kit. So the first comparison is about the size. So in human, this is eukaryotic cell, so the size of the genome is quite large. In human, the size is more than 3 billion base pair. Meanwhile, the size of the mitochondrial genome is quite small, because normally the size of the genome of the prokaryotic cell is relatively smaller than the eukaryotic ones. So the size of the mitochondrial genome is small, just more than 16,000 base pair. The number of DNA molecule, so in in the nuclear genomes, it in a somatic cell is contain 46 chromosome equivalent to 46 molecule of DNA associated with histone protein. Okay, and in mitochondrial genomes, in one mitochondria is have only one molecule of circular DNA, but because in one cell normally is have from 4,000 to 6,000 mitochondria. Okay, someone is late, it's not a problem. As I was informed from the GED, so my course is still online. So, okay. Anyway, come back to our lecture. So in one cell, because one eukaryotic cell will have from 4,000 to 6,000 mitochondria. So it corresponds to in one cell, you will have from 4,000 to 6,000 versions of mitochondrial genomes. Next comparison is about number of genes encoded. So according to the Human Genome Project, in a human genome, a normal individual will have around 25,000 genes. And gene is meaning coding for protein or other coding RNA. And in mitochondrial genomes, the mitochondrial genome will have 37 genes. Among those, only 13 was coding for a protein. The others, 22 is coding for tRNA, and 2 is coding for the subunit of ribosome RNA. So the gene size of the nuclear genome is quite large, since it's also including also the intron, which is take a large proportion but not coding for protein. Meanwhile, the mitochondrial genome, most of them was coding for protein. So in mitochondrial genome, the average size range of one gene is around 450 base pair only. The intron is nuclear genome, it is not intron. And in the intron in the nuclear genome is frequently found, and it takes a large proportion of the nuclear genomes. Meanwhile, in the mitochondrial genomes, there's no intron in the gene. So we have only the D loop, I mean the hypervariant 1 and 2 is the only reason that not coding for anything. Okay. The percentage of coding RNA, so in nuclear genome, it's only 1.5% of the genome is coding for protein, and another 1% is coding for ribosome RNA, tRNA, non-coding RNA, and other stuff. So just around 3% of the nuclear genome is coding for something. Meanwhile, a large proportion of the mitochondrial genome was coding for protein or ribosome or tRNA. Only the D loop is coding for nothing. The codon usage. So in the nuclear genome, they use the universal genetic code of the formula, the popular genetic code that we use today. Meanwhile, the mitochondrial genome, because it's an Asian prokaryotic species, so it has some unique feature. For example, the UAU is coding for methionine. The TGA in universal code, TGA is coding for stop codon, but in mitochondrial genomes, TGA is code for tryptophan, and so is others unique features. Associated protein in nuclear genome DNA is associated with histone protein to form chromosome. Mitochondria is not associated with histone, but with others protein, like we have discussed last week in the prokaryotic genomes. Okay, and the other stuff. So this is the summary of the key concept. The things that important for this chapter. Of course, maybe I have missing for the other important stuff for the chapter. So if you find out there's something important I'm missing, please add it for me. Okay. So in this chapter, you should remember about the structure of DNA. What is the nucleotide composed of? It's composed of three main parts we have discussed last week. And DNA, the two strands are complementary links to each other for the Chargaff rule. These are also discussed last week. What is differences between DNA and RNA in the structure and from the structure to their function? And what is differences between the genomes of the eukaryotic cell and the prokaryotic cell? Okay, and so on. What is the noticeable information we need to remember from this table? Okay, so if you find out that this table is too hard to remember, just remember that the nuclear genome is carry over characteristic features of the eukaryotic genomes, and the mitochondrial genomes is having all the characteristic features of the prokaryotic genomes. That's it. Okay. Okay, so do you have any question on chapter 1 before going to chapter 2? Right.
Professor, I can't find the mitochondria part in the slide that is on Moodle.
You can find there?
There is no, that part isn't exist in the slide.
Okay, so I will add this part in the slide on Moodle. I will do it tomorrow. Okay.
Okay, thank you.
You're welcome. Okay, next chapter. Yes, but it's still a new part, so I cannot finish it in the last week. So in next chapter, we will discuss with each other on the mechanism how DNA was replicated and what is mechanism, the differences in mitosis and meiosis. First, talking about DNA replication. Talking about DNA replication, we have to talk about cell division and cell cycle. So the cell division is necessary for many steps when an organism was grow and develop, and when special tissues, special cell need to be replaced because there's damage. I don't know, maybe a damage or the cell was too old, so it needs a replacement. So for the cell to divide, first it have to duplicate all of the necessary material. Follows several steps that we'll discuss later. And the key is this divide to maintain all the feature of the mother cell. So first, let's watch a small animation on the development of the zygote through cell division. Okay, so after fertilization, the sperm will combine with an egg to form a zygote. In human, the zygote cell cycle is approximately 1 day, 24 hours. Okay, so that means that after 1 day, a zygote will divide in 2 cells, and after another day, 2 cells will divide into 4 cells. After another day, 4 cells to 8 cells, another day to 18 cells, and another day to 32 cells. And from the 32 cell state, it will form a 2-layer cell, at least 2-layer cell. The outside layer, which is denoted in the purple color here, will develop into the placenta. Okay, and the inner cell, which is denoted in the red colors here, will develop into the embryo. So that means that the placenta will have the same genetic background with the embryo. The genetic screening and genetic base testing to see that if the future child carry anything, any abnormalities about the genetic background can be performed with the sample taken from the placenta. Those placenta have the same DNA background with the child. Okay. Thực tế là mình xin giải thích một chút bằng tiếng Việt là sau này bạn nào có làm ở các bệnh viện thì sẽ có biết cái dịch vụ này. Là ở một số bệnh viện hiện nay người ta đã phát triển cái soi cái sự phân bào, cái sự bất thường trong phân bào lúc ban đầu, ví dụ như là những cặp vợ chồng nào làm thụ tinh nhân tạo và người ta soi trứng thì bình thường một cái chu kỳ tế bào của cái hợp tử là khi mà chính kết hợp với tinh trùng hình thành nên cái tế bào đầu tiên gọi là hợp tử, đúng không ạ? Thì là cái chu kỳ tế bào đầu của một hợp tử bình thường sẽ là khoảng 24 giờ. Có nghĩa là sau 24 giờ, tức là sau một ngày thì từ một tế bào sẽ phân chia thành 2 tế bào, đúng không? Sau 24 giờ nữa thì phân chia thành 4 tế bào, sau 24 giờ nữa thì thành 8 tế bào, thành 16 tế bào như thế. Thì các giai đoạn phôi 3 ngày và phôi 5 ngày, phôi 3 ngày là ở cái giai đoạn 2 mũ 3 là bằng 8 tế bào, phôi 5 ngày là bằng ở giai đoạn 2 mũ 5 bằng 16 tế bào ấy. Thì người ta có thể là dự đoán được cái phôi này có bị dị tật không, để người ta cấy vào. Ví dụ như là cặp vợ chồng nào thụ tinh nhân tạo thì người ta sẽ làm một lúc mấy phôi, và các bác sĩ sẽ dựa trên cái phôi nào mà khỏe mạnh nhất, không chứa ít những cái bất thường nhất để mà chọn mà cấy vào tử cung người mẹ. Thì thường là người ta sẽ chọn những cái phôi khỏe mạnh dựa trên là scan xem là cái chu kỳ phân bào của cái phôi này nó bình thường không. Ví dụ là chu kỳ bình thường là khoảng 24 tiếng phân bào một lần chẳng hạn, nhưng mà có một cái phôi nó chỉ khoảng 16 tiếng nó đã phân bào rồi, hay có những cái phôi đến 28 tiếng nó vẫn chưa phân bào, thì người ta thấy là ở những cái phôi này thì thường tần suất xảy ra đột biến gen ở cái phôi, có nghĩa là phôi này nó không khỏe mạnh, nó rõ ràng hơn. Hoặc là có một số phôi nó phân bào mà nó không đều, ví dụ như là sau 24 tiếng thì một nửa tế bào phân bào rồi, một nửa tế bào lại chưa phân bào, ví dụ dẫn thành ví dụ như sau 26 tiếng lại dẫn thay vì 8 tế bào thì lại thành là 5 tế bào chẳng hạn, có nghĩa là có một nhóm tế bào nó phân chia sớm hơn các nhóm tế bào còn lại, thì cũng ok, thì nó cũng dẫn đến cái việc đó. Thế còn ở đây có một bạn sinh viên vừa hỏi về việc khám thai định kỳ, thì thực ra khám thai định kỳ nó hoàn toàn khác. Ở chương 4 thì mình sẽ nói sâu hơn về các cái khám dị tật ở thai nhi, nhưng mà khám thai định kỳ thường là ví dụ như người nhà các bạn đi làm ở bệnh viện là người ta chỉ có làm siêu âm thôi. Làm siêu âm thì là khi mà hình thành cái phôi đấy, hình thành nên đứa bé thì người ta sẽ soi, ví dụ siêu âm độ mở da gáy để xem là có bị đau không, hoặc là nó có dị tật gì bất thường về xương, về hình dáng không. Nhưng mà siêu âm là dựa trên cái hình dạng 3D của cái bào thai. Còn ở những cái giai đoạn này là giai đoạn rất là sớm, thường là nó chỉ là một nhúm cái tế bào thôi, thì siêu âm ở cái giai đoạn này nó không có nhiều ý nghĩa, nó chỉ là một cái phôi thôi, thì không thể nào biết được là cái phôi đấy nó có bình thường hay không. Thì người ta phải dựa trên những công nghệ khác, ví dụ như là hình thái phôi, là ví dụ như là genetic screening, hoặc là dựa trên hình thái phôi xem là cái độ phân chia tế bào để xem xem là nó có cái chu kỳ tế bào nó có gì đó bất thường không, để mà dựa trên đấy là xem cái phôi có phát triển một cách khỏe mạnh không. Đấy là thông tin bổ sung thêm cho các bạn. Okay. So, nhân thể hỏi thêm là các bạn có biết là cái tuổi thai tính như nào không? Có ai biết tính tuổi thai được tính như nào không?
Tuổi thai tính từ theo nhớ lại là
Là như nào nhỉ?
ngay đầu của cái nguyệt cuối của mẹ.
Đúng rồi, rất chính xác. Tuy nhiên một cái quan điểm nhầm lẫn khá là phổ biến, đó là tuổi thai được tính từ khi là tinh trùng bắt đầu gặp trứng, nghĩa là khi hợp tử được hình thành. Thế thì cho đến bây giờ thì vẫn có nhiều người không chuyên, người ta nhầm lẫn cái đoạn này. Thì thực tế là khi mà các bạn đi khám thai, khi mà siêu âm các thứ ấy, thì bác sĩ đưa ra kết quả là tuổi thai được 4 tuần hay 8 tuần hay bao nhiêu đấy. Thì thực tế là cái thời gian này là được tính từ khi cái tế bào trứng non ấy chuẩn bị bước vào bắt đầu bước vào quá trình nguyên phân để giảm phân để tạo thành tế bào trứng trưởng thành. Ví dụ như là các bạn biết là ví dụ một tế bào sinh tinh thì qua quá trình giảm phân cho ra 4 tinh trùng khỏe mạnh, đúng không? Một tế bào sinh trứng thì qua quá trình giảm phân chỉ cho ra được một tế bào trứng thôi, vì là cái trứng thì yêu cầu cái lượng tế bào chất nó nhiều, nên là tất cả những cái nhân kia ba cái nhân kia bỏ đi hết, chỉ giữ lại một cái nhân mà nhiều trứng nhất thôi, cái trứng mà nhiều tế bào chất nhất thôi. Thì cái quá trình mà trứng non bắt đầu hình thành, xong rồi trưởng thành, xong nó rụng xong rồi qua quá trình di chuyển từ vỏ trứng đến tử cung gặp tinh trùng, thì từ ngày nó thông thường nó rơi vào khoảng 2 tuần. Cho nên ấy là cái tuổi thai thông thường là nếu mà tính tuổi thai thì nó sẽ sớm hơn cái thời điểm mà trứng gặp tinh trùng khoảng độ 2 tuần. Đấy, cho nên một số thông tin để bổ sung các bạn, không có sau này ông nào đi đưa vợ đi khám thai, đúng không, tính ngược lại thầy bảo ô cái gì đấy nó sai sai, bảo hôm đấy mình đang đi công tác mà nó không đúng, đúng không? Ok. Okay, so talking about cell division, we will discuss about mitosis and meiosis. So this is the two process that you have studied in high school. So mitosis is the cell division in most of the cell, the somatic cell, and the germline cell, gamete produce cell in the early state. And the meiosis is the cell division happen in the gamete produce cell, mature cell, in the mature egg cell and sperm cell. So in mitosis, one cell will passing through one time the DNA was duplicate, and one time the cell was divided. So from as a consequences, from one mother cell will provide two daughter cell with their genomes exactly like the mother cell. Meanwhile, in the meiosis, from one mother cell been through one time the DNA was duplicate, and two times the cell was divided to provide four daughter cell with the number of chromosomes was decreased by a half compared to the mother cell. So the DNA replication was can divide into two main phases. One is the interphase, the preparing phase, which is can categorize into three phase: G1 phase, the S phase, and G2 phase. G1 phase is the preparation for all the enzyme or the material necessary for the DNA replicated. The S is for the DNA was replicated. The G2 phase is when the cell checking for the size the enzyme, the DNA was fine, everything was fine, preparing for the mitosis. And the interphase is also the longest phase in the cell cycle. As for example, for one cell cycle is 24 hours, the interphase will take around from 18 to 22 hours. And the mitotic phase actually happen quite shortly and can divide into 4 phases. And our process DNA replication is mostly on the S phase. So in the beginning of 19th century, in the beginning of 20th century, scientists still wonder on which model DNA would replicate. So they provide 3 main models. The first one is semi-conservative model. That means that from one parent DNA, which is denoted in the blue color here. So each template, each strand will be used as a template to synthesize one newly DNA strand. The new DNA strand was denoted as the purple color here. Okay, so after first time replicate, so it's make 2 DNA strand with the sequence exactly like the parent DNA. Each double strand DNA will have 1 strand is have the origin from the parent, and the other one is newly synthesized, and so on. On the conservative model, scientists provide that demonstrate that the 2 there will be 1 double strand DNA will have both 2 the DNA strand is parent original, and the other entirely double strand DNA will newly synthesized. And the last model was deeper-safe, which is depend on the region. One part of DNA will from the parent, and the other part is newly synthesized. So until 1957, the experiment designed by Michelson and Starr have been proved that the DNA was synthesized follow the semi-conservative early model. So the experiment is performed like this. So for the control, the bacterial E. coli will be raised on the medium contain the nitrogen 14. So in nitrogen, we have 2 radioisotope is N14, which are corresponding to 99.9% of the nitrogen we can find in the environment. And the other radioisotope, quite rare, is N15. So in the control, they rose the E. coli in the N14. When they extract the DNA, they find out that both 2 DNA strands is composed from the N14. So the first experiment, they take E. coli grow in for many generations in the medium containing the radioisotope N15. After a few generations, they found out that all the DNA now composed from the N15 radioisotope. After those, they pick up 1 colony and raise this E. coli strain in the normal medium contain regular nitrogen, that means the nitrogen 14 isotope. And after each generation, after each cell division, they will extract the DNA and try to calculate the density of the DNA molecule. So after 1 generation, they find out that there is only 1 band when they make a centrifuge. Centrifuge, that means they put it in the centrifuge force, and which molecule will have a higher density when we take it in the lower position of the tips. So after one generation, they find out that only two bands there, and because there's a combination between the N15 and N14. And after two generations, they have found out there's two bands of DNA of the E. coli. The 15 and 14 is still there, but there's a band for lighter N14 and N14. So after each generation, the N14 was increased. After each generation, the combination of N15 and 14 was decreased by a half. So based on this method, they have proved that the DNA was replicate inside E. coli, follow the semi-conservative replication. So let's watch a small animation on to demonstrate the experiment of Masterson and Stahl.
The Messelson and Stahl experiment provides evidence for semi-conservative replication.
Sorry. Okay.
The DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
So on the semi-conservative replication, so from the double-strand DNA, some special enzyme called helicase will cut down the hydrogen bond that link between the two strands of the DNA double strand. So from this each template here, each strand of mother DNA strand here will be used as a template for the DNA polymerase to synthesize the new DNA strand. Follow the Chargaff rule. A is linked with T, G is linked with C. So as a consequences, from one mother double-strand DNA will produce two daughter double-strand DNA with the sequence of DNA are the same with the first with the mother DNA, and each DNA daughter cell, each DNA molecule, each daughter DNA molecule will have one strand was parent original and one other strand was newly synthesized. So DNA replication as the other copy process in the cell always have mistake. I mean DNA and mistake can cause DNA mutation, and we can discuss about it later. But the main proportion participate in this process is the number of enzymes participate in this process is nearly 100 enzymes. But for now, we just focus on the main part, the main enzyme. So first is we have the helicase, which it cut down the hydrogen bond linked with the two double-strand DNA. We have binding protein that's stabilize the separate strand. We have primases at the primer in the template strand. DNA polymerase, the main part here, will start synthesize the newly DNA strand from the primer RNA primer. The isonuclease will remove the primer and add the DNA BNTP to form a DNA oligo from at the position of the RNA primer. And finally, we have ligase when join the discontinuous fragment together. So first, from the double-strand DNA, I will share my screen. So DNA replication. So first we have double-strand DNA in here, okay? And this two is linked with each other by hydrogen bond. So this is the double. This is the hydrogen bond. So the first enzyme here, the first enzyme we have helicase that cutting down the hydrogen bond that link the two strands of DNA. So we first helicase. So after it's cutting down, we have two separate DNA strands here. The next part is stabilized protein. Binding in here to make
Excuse me,
sure, yeah.
if there is not if the binding proteins isn't present, what happens? What will happen?
Well, because the hydrogen bond is really performed together, because if we don't have this hydrogen bond, maybe these two strands at some point will come back and binding together again, because these two hydrogen bond it can come back and join together. And when it join together, there's no space for the DNA polymerase to come and binding and start to synthesize the nuclear DNA strand, because this process needs space for so the other complex can come and start to synthesize the nuclear DNA. So if
Okay, so is it that they are separate? Like those proteins keep these strands separate?
Yeah, it keeps the two DNA separate, so to maintain the space, so the other complex can come and binding in there. Okay. So this protein. Đang chơi điện tử, tắt mic đi nha. Đánh nhau cứ bùng bùng. In a single strand. Okay. So next enzyme we have primase here. We will add the primer into the template, DNA template. So we have primase. Let's put it in green. Primase. RNA primer. And remember, this primer is RNA, okay? It's not DNA. Okay, so this is this one is RNA prime. Okay. So in the next step, the DNA polymerase will come and binding in the primer and start to synthesize the DNA strand from the direction 5' to 3'. So what is the 5' to 3'? You have to come back to the last chapter, the chapter last week to remember. If you follow, forget what is 5' and what is 3'. So I have 5' here and 3'. So because this strand is complementary with the upper strand, so we will have 3' here and 5' here. It's parallel, remember? So in here will be the 3' and DNA polymerase. So our main core here, sorry. DNA polymerase. Start. So here the DNA polymerase synthesize the new DNA strand from the primer position. And in here, please note that DNA polymerase only work from 5' to 3' direction, not
So, so the polymerase slides from the 3' to 5' and synthesizes from 5' to 3'. Is that right?
yeah. So wait me a minute. So in here, because this strand, the 5' here and the 3' here, so the position of 5' here, so and the 3' is in here, okay? So it start to binding here and start to synthesize from 5'. This is the 5' of the and start to synthesize to 3'. Okay, and in the in the other strand, because in this strand the direction was totally opposite, so it must have start to synthesize from the way around. So in here 5' here 3', so the 5' was in here and 3' was in here. And because DNA polymerase only working from the direction of 5' to 3', so it will start to synthesize like this. Of course, based on the direction, because the helicase continue to break the DNA right here, so as a consequences, there's many traces to need to synthesize. So in this strand, in this upper strand, the DNA, the new DNA strand was is synthesized continuously. So we call it is the leading strand. And in this down strand, the new DNA is synthesized discontinuously. So we call this strand is the lagging strand. And this fragment here, this continuous fragment
Are they called Okazaki fragments?
yeah, thank you. Okay, this one, and this one is called Okazaki fragment. Okay. So next part, we will have in next part we will have a exonuclease here on someone called the DNA polymerase 1. It's the same. We'll remove the RNA primer and change the DNA change the RNA primer by the DNA oligos. So we have a guide, enzyme. Let's call this one. Okay. This is called exonuclease. This one will come and delete all the RNA primer, all the RNA primer that denote in green here. You can see it, right? Delete the RNA primer and insert the DNA to it. Okay. Where I wrote and write. Okay. Primer. With DNA oligos or DNA fragment, or it's the same. It's the same meaning. So it erases all the RNA primer and replace it with DNA. So we have the strand here is DNA. We have this fragment here is also DNA. We only have the gaps between those. The lagging strand is also consequences of the DNA replication. So as you can see here, after synthesize, after DNA replication, we still have 2 newly DNA strands. Okay. And the final one is ligase. Final one, we have ligase. Let's join up. Okazaki fragment. By sealing the sugar and phosphate backbone. The DNA strand. So far. Finally, we'll have, yeah.
If they're if ligas doesn't exist, then what will happen to the DNA?
If DNA ligase doesn't happen, so we will have this. I will open a new pane, maybe. You want to save this? Maybe I will save this in the desktop. Replication. So if DNA ligase doesn't exist, we will have something like this. Okay. So okay, so here's the helicase. But in here we will have one, and this is a primer erased by DNA. We have a seal here that's not the sugar phosphate bond here does not link, and we have so many discontinuous like this fragment. Okay.
Okay.
So we did the ligase will seal the sugar and phosphate backbone. Do you remember in chapter 1 we will have discontinuously like this instead of this. Okay. Okay.
Okay.
Yeah. Great. Chapter 2. So as a consequences, finally, we from one parent strand, we will have two daughter strand with the sequence exactly like each other. And let's watch a short animation to demonstrate how DNAs will replicate inside equalized cell. Okay.
Cells like these prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, 2 identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
What will happen if the single-strand DNA is coiled? I think I have explained this in the previous chapter. So in the supercoiled state, there's no space for the DNA. So the supercoiled state is good for the cell to divide, but it's not good for DNA replication since the supercoil did not create a space, so the other complex to binding in. Okay. Ok, giải thích lại bằng tiếng Việt là để cho các cái quá trình sinh học, ví dụ như là DNA replication diễn ra một cách bình thường, thì toàn bộ các cái ví dụ DNA replication thì thực tế là nó là một cái complex rất nhiều protein, nó đến nó rất nhiều cái enzyme, nó đến nó tiến hành quá trình sao mã DNA này, thì để các cái enzyme có khả năng đóng vai trò trong việc sao mã DNA đến, thì cái phần tử DNA đấy nó phải ở một cái vị trí không gian nó mà các enzyme nó tiếp cận được. Trong cái trạng thái mà supercoil, nghĩa là DNA nó xoắn cực đại ấy, thì các cái enzyme nó không thể nào nó tiếp cận được vào cái không gian đấy. Ví dụ như là ok, thì ví dụ đây là sợi DNA, thì muốn cái quá trình này diễn ra thì nó phải có một cái.
Teacher, you haven't shared screen.
Sorry. Ok. Đây, ví dụ đây là sợi DNA đúng không ạ? Thì nếu mà cái đây ví dụ như là đây là toàn bộ các cái enzyme đóng vai trò của công việc replication. And enzyme I use the plural. Thì nó phải có cái không gian 3D, có không gian để mà toàn bộ những cái enzyme này có thể tiếp cận và bám được vào cái sợi DNA đấy, thì cái quá trình sao mã DNA nó mới diễn ra được. Còn trong trạng thái mà DNA nó lại đang xoắn một cách cực đại như này đấy, thì ví dụ nó muốn sao mã cái đoạn này chẳng hạn, cái đoạn mà tôi đánh dấu màu đỏ này, đoạn mà tôi đánh dấu màu đỏ đây, thì cái phức hệ DNA replication enzyme này làm sao nó bám được vào đây để mà nó tiến hành quá trình sao mã, đúng không? Thì nó phải giãn xoắn ra, đầu tiên nó phải giãn xoắn ra, hay như là ở cái eukaryote thì nó cũng phải giãn xoắn mà nó tháo bớt các cái protein histone ra, thì nó mới có không gian để mà DNA replication enzyme, hay là sau này các bạn học về phiên mã thì cái nhân tố phiên mã có không gian để mà bám vào để mà mới xảy ra các quá trình phiên mã hay là sao mã được. Chưa? Any question on this? Okay, so next is about mitosis. So mitosis is happen mostly on the somatic cell, and also in the gamete cell in the early state when it's not mature yet. So also mitosis can be used for reproduction in some species as asexual reproduction. So mitosis was composed by 5 stages, some also the 4 states, because interphase is the preparation state. So in the interphase, the cell was prepare everything for the cell division. The DNA was duplicated by us. All the enzyme or material or the plasma will prepare for the cell division. And normally the interphase was the longest phase in the cell cycle. So to start the mitotic phase, the phase in prophase, the nucleus membrane will disappear and the centriole will start to move in the 2 sides of the cell and start to form the microtubule here, and start to the microtubule will attach into the centriole centromere of the chromosome. Start to meet in the metaphase, the chromosome start to arrange in the middle of the cell Some call this is a metaphase plate, that the plate into the equator of the cell here. And the double chromosome will gather in here until the anaphase, the microtubule will start to separate the double chromosome together and separate each part of each chromosome into one side of the cell. And in the final state, telophase, the cell membrane will form again, and the cell membrane was divided from one mother cell to two daughter cells with the genome exactly like the mother cell. So let's watch us animation.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
So during DNA replication, the process can provide error, and error during DNA replication causes DNA mutation. According to a report in scientific report, a virus, DNA virus, that means a virus with the genetic material with DNA, have a mutation rate from about 10 power by minus 6 to 10 power minus 8. That means in 1 million to 100 million base pair, it will cause one mutation. The virus with RNA as a genetic material is much have a mutation rate much higher, because normally the RNA is less stable than DNA. So the mutation rate in RNA virus is from in 1,000 base pair to 100,000 base pair. They want genetic mutation. In human, the mutation rate is about 100 million base pair per one mutation. But because in higher high level organism like in human, we have the DNA repair or DNA mutation repairing system. So is the successful rate of this system is around 99%. So it will decrease the mutation rate 100 times. So the mutation rate after with the DNA mutation is still around 1 per 10 power by minus 10. That means in 10 billion base pair, we will have one mutation with the genome size of 3 billion base pair. So that means in around 3 cell cycle, we will have one type mutation. Most of the DNA mutation is randomly, and depend on the position of the mutation can be really, really harmful or totally benign to the carrier. So in the animation, they provide three to provide a very interesting term called the cell cycle checkpoint. So the cell cycle checkpoint is the position in the cell cycle that to maintain to make sure that if the cell cycle was happening normally. So we have three main cell cycle checkpoints. One is after G1 phase to check for if all the DNAs were fine to start going to phase S. And if it fails to the GN checkpoint, it will go to the resting state. I mean, the DNA stop synthesis. And in some case, it can go to the apoptosis. That means the cell death follow programming. The second checkpoint is G2 checkpoint. It check for after the S phase, the DNA replication was happened normally, and all the enzyme or the material necessary for the mitotic state was enough for the cell division. And the last one is the M checkpoint, is the checkpoint after phase S to make sure that all the cell was divide normally. So that's a good checkpoint.
Cell growth and division are divided into phases. Cells grow during the G1 phase, and the genome replicates during the S phase. During the G2 phase, cells continue to grow and prepare for cell division. During the M phase, mitosis occurs. Cytokinesis, C, is the phase when the cytoplasm divides, creating two daughter cells. Three principal checkpoints control the cell cycle in eukaryotes. The G1 checkpoint makes the key decision as to whether the cell should divide, delay division, or enter a resting stage. The G2 checkpoint assesses the success of DNA replication and triggers the start of the mitosis, M phase. If this checkpoint is passed, the cell initiates the beginning of mitosis. The accuracy of mitosis is assessed at the M checkpoint. This checkpoint occurs during metaphase and triggers the exit from mitosis and cytokinesis and the beginning of G1. At the G2 checkpoint, cyclin-dependent kinases, CDKs, phosphorylate histones and proteins that carry the cycle past the checkpoint into mitosis. During G2, the cell gradually accumulates G2 cyclin, also called mitotic cyclin. The cyclin binds to CDK to form a complex called MPF, mitosis promoting factor. When the level of MPF exceeds the threshold necessary to trigger mitosis, the G2 phase ends and mitosis begins. One of many functions of MPF is to activate proteins that destroy cyclin. As mitosis proceeds to the end of metaphase, CDK levels stay relatively constant, but G2 cyclin is degraded, causing progressively less MPF to be available and initiating the events that end mitosis. After mitosis, the gradual accumulation of new cyclin starts the next turn of the cell cycle. The G1 checkpoint is thought to be regulated in a similar fashion. The level of G1 cyclin increases and associates with cyclin-dependent kinase, CDK. Eventually, a threshold ratio that triggers the next round of DNA replication is reached. The cyclin is degraded and the cycle begins again.
Okay. So the next part on the DNA mutation and related to cancer, I think I will save it for Thursday. Do you have any question for today's lecture? Please remember that from tomorrow we will have a bioinformatics training course, so please be on time, okay?
Okay, so excuse me, can you send the whole like the whole slides, because my house lost electricity.
Your house lost what? I'm sorry.
Electricity.
Okay, so you want the slide?
Yes.
I will upload the slide again on the Moodle. So you can go. You don't have your Moodle account?
Yes, I've already got the Moodle account, but the slides that they seem to be different.
Different. Okay. So I will update the slide on the Moodle account. So don't worry. So maybe I will do it tomorrow. So please check the Moodle on the weekend to make sure that you have received the proper slide. Okay.
Okay.
Okay. Thank you. Any other question?
No, not right now.
Okay. Thank you very much. So since you have no more question, I want to thank you for your attendance and see you in Thursday.
See you, Professor.
Thank you. See you.
Bye.
Thank you.
Bye, Professor.
Bye, Professor.
Thank you. See you.
Okay, see you later, teacher.
See you.